Hello there, and welcome to how to interpret my Plasmidsaurus sequencing results. This video will be a brief walkthrough on interpreting your linear PCR results directly on the Plasmidsaurus website. Once your results are ready, you're going to receive an email that looks a little bit like this one. This summary report gives you a quick snapshot of the linearized feature maps of all of the samples in your experiment. If I click on the View Results button, it's going to take me to the website order page. Now, here on the summary table, you can see we've got a lot of overview information, and you can also see that I've uploaded a reference for each of these. This can be done by clicking on Manage References, and then clicking the Upload Files button and finding the file on your system, or you can simply drag and drop into this box. The supported file types are GenBank, Fasta, and Snapgene. The great thing about that is if you do upload a reference, we will automatically align your consensus sequence to that. And so you can see here that if it matches perfectly, you get this happy looking dinosaur. And if it has one or more mismatches, then you get the dreaded chicken. Everything here is clickable and I can jump around. So if I go to number 1, you can see that we've got this per base information, the amino acid prediction. I can zoom around between all the different parts. And we've also got the per base support as well. Now, if we scroll down to tube number 2, we can see that, we have got a mismatch. So if I click it, it's going to take me directly to that. And you can see in this case, we have got a SNP. So in the reference, we've got an A, but our sample has got a G. And if I go up to the per base information, you can see that that G is very well supported. Previously, we were making phenylalanine, but now we're making a lysine. Phenylalanine is large, hydrophobic, and non-polar, whereas lysine is small, positively charged, and polar. So there's a good chance that this may have an impact on function. If I keep scrolling, you can see that in tube 3 we've got a perfect match to reference, which is great, and you can see that we will also annotate things that aren't annotated in that reference. If I look at tube number 4, again, you can see that we have got a mismatch. And again, I can click on it and that will take me directly to it. So you can see in this example that this is probably a bigger problem. So we previously had a stop, but we have got a SNP again. So this G has been changed to a C, which again is very well supported by the underlying data. And so instead of a stop, this is now a serine. And of course that means it doesn't stop making protein. So there's a good chance that you will have to remake this. And this is a great example of why we say you should sequence everything, because you just drop this in the Dropbox, and by the time you come into the lab the next day, we've sequenced this, you've got this result, and you can look at this and say, this is misformed. I need to make it again, or I need to use a different plasmid, or whatever it is that you need to do, rather than spend, days, weeks, months troubleshooting an experiment that was never going to work. Okay, well, thank you so much for watching. I will see you next time.